Read the introduction to question 2 before you proceed to the rest of the video. For question 2, this is what's happening in the experiment. In the test tube, there is 5 cm cube of buffered solution. It's a solution that is made to a certain pH. There's 5 cm cube of amylase and 30 cm cube of starch. For each of the other 7 test tubes, the only difference would be the pH of the 5 cm cube buffered solution. Please take note that the amylase is required in small amounts because enzymes remain chemically unchanged and therefore can be reused. So the other portion of the experiment is, this is what's happening when they start the time. Every 30-second interval, they take 1 cm cube and test for starch until the time they get a yellow or brown, which indicates that starch has been digested. So this is another experiment to show that food test is used as an indication that digestion, in this case enzyme action, has taken place. So at the start, when there is starch, that's because starch has not been digested. Whereas at the end of the time that it becomes yellow, it means starch has been digested. So let's take a look at the question. The variable that is being tested in this experiment is definitely pH. And the 2 conditions that you have kept constant, one is already given in the experiment, and that is the volume. The total volume of the reagents is kept constant at 40 cm cube. So volume of reagents because it is an all-inclusive term for whatever is inside a test tube. Okay, the second one, which is not written in your question, is of course temperature. C. From the results, what is the optimum pH of amylase? And explain your answer. So it's clear that the answer needs to be given from the results, that means from the graph. So our usual graph is the rate of enzyme action or the rate of digestion or reaction over pH. Okay, so speed and time are inversely proportional, and therefore here the faster the time, the shorter the time taken. So the pH is definitely pH 7. To explain your answer, when we explain answers, we explain in 2 ways. We explain using a biology concept, or we state how we get the answer from data. So in this case, we use the answer that is in your data, because it takes the shortest time at pH 7. It takes the shortest time to convert starch completely. Okay. In the diagram below, show the amount of substance, how the amount of substrate varies during the reaction. To answer this question, we need to understand that over time, the amount of substrate decreases. Okay, so you started with 30 cm cube of starch. Okay, so over time, as amylase digests starch, then it will go to 0 cm cube because all the starch has been digested. So as long as your graph looks like it's coming down, then it is a valid graph. Whether it's straight or curved, a sketch is an estimated drawing of the graph. So this is the answer for question number 2.